Have you ever wondered how genetic discoveries are transforming our world and the way we understand life itself? From the moment that Watson and Crick unlocked the structure of DNA back in 1953, we've been on a relentless journey of discovery, a journey that's revolutionizing everything from healthcare to forensics, from agriculture to anthropology. Genetic discoveries have given us the power to cure diseases, solve crimes, grow more resilient crops, and even trace our ancient ancestry. But at the heart of all these breakthroughs are tiny molecular structures known as nucleotides. These are the unsung heroes of genetics, the fundamental building blocks that make up our DNA and RNA, the blueprints of life itself. Nucleotides are like the letters of an intricate genetic alphabet, spelling out the instructions that guide every process in our bodies. To better understand these impacts, we must first delve into the fundamental building blocks of life, the nucleotides. At the heart of every organism, there are 5 nucleotides that form the language of life. These simple molecules, adenine, guanine, cytosine, thymine, and uracil, are the basic building blocks of our genetic code. They are like the letters of an intricate alphabet, coming together to spell out the instructions for life. Let's start with adenine, a crucial component of both DNA and RNA. It's a purine nucleotide, meaning it has a 2-ring structure. It's a bit like a master key, capable of pairing with thymine in DNA and uracil in RNA. Next up, we have guanine, another purine nucleotide. It's a bit like adenine's sibling with a similar 2-ring structure. But guanine has a different dance partner. It pairs up with cytosine. Speaking of cytosine, it's a pyrimidine nucleotide, having a single ring structure. It's a bit like a puzzle piece, always seeking its perfect match, which in this case is guanine. Now, let's talk about thymine. This pyrimidine nucleotide is exclusive to DNA. It's the perfect match for adenine, forming a pair that is crucial for the structure of the DNA double helix. Finally, we have uracil, another pyrimidine nucleotide. But unlike thymine, uracil only appears in RNA, where it forms pairs with adenine. These 5 nucleotides are not just simple molecules. They are like dancers, forming intricate pairs that help to shape the double helix structure of DNA and the single-stranded structure of RNA. This dance is not just beautiful, but purposeful. It allows the genetic code to be read, copied, and translated into the proteins that drive life's processes. Consider them as the words in the story of life. They are the language that every cell in our body understands. They spell out the instructions for growth, development, function, and reproduction. They are the code that makes us who we are. These nucleotides come together to form the DNA and RNA, the genetic codes of life. They are the alphabet of life, and understanding their language is key to unraveling the mysteries of the living world. DNA and RNA, these 2 molecules hold the blueprint of life. These biological architects, DNA or deoxyribonucleic acid and RNA or ribonucleic acid, are made up of nucleotides. Each nucleotide comprises a sugar, a phosphate group, and a nitrogenous base. DNA houses 4 types of bases, namely adenine, thymine, cytosine, and guanine. In RNA, thymine is replaced by uracil. DNA, the mastermind behind life as we know it, is a double helix, 2 strands running in opposite directions. The strands are connected by pairs of nitrogenous bases, where adenine always pairs with thymine and cytosine with guanine. This pairing is the language of life, dictating every biological process. DNA is also aided by proteins called histones, which the DNA wraps around to form a complex called chromatin. This structure allows the DNA to fit neatly inside the nucleus and plays a crucial role in gene regulation. The secondary structure of DNA is this iconic double helix, while the tertiary structure refers to the supercoiling of the DNA molecule into a form. This supercoiling is essential for DNA replication and gene regulation, contributing to the overall diversity of life. On the other hand, RNA, though similar to DNA, has its unique characteristics. It's usually single-stranded and plays a crucial role in protein synthesis. It reads the instructions coded in DNA and translates them into proteins, the workhorses of the cell. RNA also exhibits versatility in form and function. There are different types of RNA, including messenger RNA that carries genetic information from the DNA, transfer RNA that brings amino acids for protein synthesis, and ribosomal RNA that forms the core of the cell's protein factory. Additionally, small non-coding RNAs like microRNAs, siRNAs, and piRNAs offer a variety of functions in the cell, including gene silencing, regulation of gene expression, and defense against viral infections. It's important to note that while DNA maintains the genetic continuity of life, RNA ensures the diversity and adaptability of life. They are the yin and yang of working together to create, maintain, and evolve life. So while DNA and RNA might seem like just a bunch of letters and shapes, they are indeed the architects of life, building and rebuilding, coding and recoding, persistently and tirelessly to facilitate the symphony of life we witness every day. DNA and RNA, in their unique structures, facilitate the diverse and complex life forms we see around us. Now, let's talk about proteins, the workhorses of the cell. Proteins are complex molecules that perform a wide array of functions within our bodies. They are responsible for building tissues, aiding in cell repair, and facilitating important bodily processes. But what gives proteins this versatility? It's all in their structure. Proteins are made up of building blocks called amino acids. There are 22 different types of amino acids that can combine in countless ways to create a protein. Each amino acid has a unique character and specific role, contributing to the function of the protein it forms. These amino acids include the likes of glycine, the simplest of them all, to the more complex ones, such as tryptophan. Now, the structure of proteins can be divided into 4 levels. The primary structure is the sequence of amino acids, like letters in a sentence. The secondary structure is formed by hydrogen bonds between the amino acids, creating patterns such as alpha helices or beta sheets. These patterns fold and twist to give rise to the tertiary structure, the overall 3-dimensional shape of the protein. Finally, the quaternary structure forms when multiple protein molecules or subunits join together. These structures are not just for show, they determine the protein's function. For instance, enzymes, which speed up biochemical reactions, have a specific shape that allows them to bind with their target molecules. Antibodies, proteins of the immune system, have a Y shape that enables them to bind to harmful invaders in the body. The intricate design of proteins is a testament to nature's genius. An array of 22 simple building blocks can give rise to a multitude of proteins, each with its unique function. It's like a complex puzzle where every piece has its perfect place. Proteins, with their complex structures, perform a myriad of functions within our bodies. So, next time you enjoy a protein-rich meal, remember, you're not just fueling your body, you're also providing it with the building blocks it needs to function. From DNA to RNA to protein, this is the central dogma of life. In essence, the central dogma describes the flow of genetic information within a biological system. It's a journey that starts with DNA, the keeper of our genetic code. Through a process called transcription, DNA unwinds and serves as a template to create a strand of messenger RNA, or mRNA for short. This molecule is like a courier, carrying the genetic instructions from the nucleus to the cytoplasm of the cell. Once it arrives, the real magic happens. In a process known as translation, the mRNA works with ribosomes and transfer RNA, or tRNA, to synthesize proteins. These proteins are the workhorses of our cells, performing countless tasks that keep us alive and functioning. And so, through this elegant dance of molecular interactions, the information in our DNA is transformed into the proteins that shape us. This fundamental process is the basis for all life as we know it. So how does this genetic information get passed on to the next generation? Let's dive in. In prokaryotes like bacteria, the transfer of genetic material can occur through 3 key mechanisms: transformation, conjugation, and transduction. Transformation is when a bacterium picks up DNA from its environment. Conjugation, on the other hand, involves the direct transfer of DNA between 2 cells that are temporarily joined. Transduction occurs when a virus carries bacterial DNA from one bacterium to another. In eukaryotes like animals and plants, the transfer of genetic material is traditionally through sexual reproduction. This involves the fusion of male and female gametes, each contributing half of the genetic material to produce offspring with a unique combination of genes. This ensures genetic diversity, a critical component of evolution. However, some eukaryotes also reproduce asexually, creating offspring that are genetically identical to the parent. This transfer of genetic material is how traits are passed on and life continues. Understanding these genetic concepts opens up a world of possibilities. The applications are vast, especially in the fields of cell and molecular biology. By mapping the blueprint of life, we can gain insights into the complexities of diseases, enabling us to develop targeted therapies and personalized medicine. Imagine a world where your treatment is tailored to your unique genetic makeup, enhancing its efficacy while minimizing side effects. Furthermore, understanding genetics revolutionizes our comprehension of inherited traits. It allows us to predict the likelihood of passing on certain genetic conditions to our offspring, empowering us to make informed decisions. It's like having a sneak peek into the future. Moreover, genetic knowledge has the potential to combat global challenges like hunger and climate change. By manipulating the genes of crops, we can create varieties that are more nutritious, disease-resistant, and climate-resilient. Indeed, genetic discoveries are not just about understanding life, but also improving it. Let's delve deeper into the world of genetics. Genes, the specific segments of DNA that code for proteins or RNA molecules, influence our traits and characteristics. We humans have approximately 25,000 genes. These genes are contained within structures known as chromosomes. With 23 pairs of chromosomes, one set inherited from each parent, we are a blend of our ancestral genetic information. Now, when we talk about genotype and phenotype, we are referring to the genetic makeup of an organism and the observable traits resulting from the interaction of genetics and the environment, respectively. Alleles, on the other hand, are different forms of a gene that can occupy the same position or locus on a chromosome, contributing to our genetic diversity. Understanding Mendelian inheritance, based on the principles proposed by Gregor Mendel, allows us to classify inheritance patterns into dominant, recessive, and codominant traits. And then we have mutations, changes in DNA sequences that can be inherited and contribute to genetic variation. These mutations can be spontaneous or induced by external factors. Genetic disorders, conditions caused by abnormalities in an individual's DNA, include examples like cystic fibrosis, Down syndrome, and sickle cell anemia. Meanwhile, genetic variation, the differences in DNA sequences among individuals within a population, provides the raw material for evolution. Finally, let's not forget genetic engineering, the process of manipulating genes to achieve specific outcomes. This includes producing genetically modified organisms, or GMOs, and developing gene therapies. These concepts form the basis of our understanding of genetics and its applications. So, as we continue to unravel the mysteries of our genetic code, remember, every discovery brings us one step closer to understanding the very essence of life itself. Opening a new chapter, let's explore the yeast-2 hybrid system, an ingenious technique that has revolutionized our understanding of genetics. This molecular biology technique is a powerful tool used to discover protein-protein interactions, which are crucial for many biological processes. It's like a matchmaking service at the molecular level. Here's how it works. The yeast-2 hybrid system uses two different protein domains, the DNA binding domain and the activation domain. When these two domains interact, they turn on a reporter gene. This interaction indicates that the two proteins can bind together. This system is widely used in research, not only to identify unknown protein partners, but also to confirm suspected interactions. Imagine it as a detective, uncovering the intricate web of protein connections that underpin the functioning of cells. The yeast-2 hybrid system, with its ability to uncover this complex network of protein interactions, plays a significant role in our understanding of genetics. It's a prime example of how innovative techniques continue to push the boundaries of our knowledge, bringing us closer to unraveling the mysteries of life encoded in our genes. Now let's shift our focus to the nucleus, nucleolus, and chromosomes, the central components of human cells. The nucleus, often referred to as the control center of the cell, is the guardian of the cell's DNA. It directs the synthesis of proteins and ribosomes, orchestrating the rhythm of life at a cellular level. Nestled within the nucleus is the nucleolus, a tiny but mighty player in the cellular symphony. Its main role? Producing ribosomes, the protein factories of the cell. It's like the architect designing the blueprints for the cell's protein production. Next, we have the chromosomes, these thread-like structures located inside the nucleus of animal and plant cells. Each chromosome, like a coded message, is made of protein and a single molecule of DNA, carrying the cell's genetic instructions. Imagine them as the cell's guidebook, containing all the information needed for the cell to function and thrive. The nucleus, nucleolus, and chromosomes each play a pivotal role in the functioning of human cells. Their intricate dance contributes to the complexity and diversity of life, emphasizing once more the marvels of genetics. They are the conductors, the composers, and the musicians in the symphony of life, a masterpiece encoded in our genes. In the intriguing world of molecular biology, blotting techniques serve as unique detectives, indispensable tools for identifying precise biomolecules. Let's explore the captivating universe of blotting. We begin with Western blotting. This method, named not for geographical predilections, but in honor of its inventor, a scientist with a fondness for Western movies. Developed in the late 1970s, it was a significant leap forward for protein detection. The tools of the trade for this method are the gel electrophoresis apparatus to segregate the proteins, a PVDF or nitrocellulose membrane for transfer, and finally, specific antibodies for detection. It's akin to a wanted poster for proteins, pinpointing and studying these critical molecules. Next is Eastern blotting, a method employed to examine post-translational modifications of proteins. This technique, developed over the years, is a master of exposing the masked alterations that can drastically alter a protein's functionality. This complex process uses an isoelectric focusing gel for separating proteins based on their isoelectric points, and a variety of labeling techniques to highlight modifications. Picture it as a seasoned detective, disclosing the secrets of protein behavior. Following this, we encounter Northern blotting, a technique employed to study gene expression by identifying specific RNA molecules. Adopted in the 1970s, it uses an agarose gel for separation, a nylon or nitrocellulose membrane for transfer, and a radio-labeled RNA probe for detection. Consider it as a microphone, amplifying the voice of the genes. Lastly, we look at Southern blotting, named after its inventor, Edwin Southern, who introduced it in the 1970s. This technique uses gel electrophoresis for separation, a nylon or nitrocellulose membrane for transfer, and a labeled DNA probe for detection. It is akin to a magnifying glass, zeroing in on the unique patterns in our genetic code. These blotting techniques, each with its unique application and distinct history, are fundamental in genetic investigations. They are the detectives, the inspectors, the amplifiers, and the magnifying glasses, assisting us to decipher the mysteries of life at a molecular level. Let's plunge into the intriguing universe of biology, featuring the ELISA technique, a robust device for estimating proteins. ELISA, or enzyme-linked immunosorbent assay, is a strategy employed to quantify the concentrations of distinct proteins in a sample. Think of it as a precise bookkeeper, accountable for the protein capital in our body's biological bazaar. This technique involves an antigen-antibody reaction, with the protein of interest being seized by a particular antibody. It's like a dedicated watchman, safeguarding and ensuring that only the appropriate proteins pass through. Now let's decipher the principle of ELISA. The protein is captured by an antibody, followed by the addition of a second antibody linked to an enzyme. When a substrate is added, the enzyme triggers a color change, the intensity of which corresponds to the protein concentration. Visualize it as a color-coded security system, where the intensity of the alarm directly signifies the intruder's magnitude. However, the field of biology has also embraced the concept of multiplexing. Novel techniques like multiplex immunoassays have replaced ELISA, allowing multiple proteins to be studied simultaneously, increasing efficiency and reducing sample volume requirements. It's like having a multitasking assistant who can monitor several proteins all at once. Data analysis in ELISA and multiplex immunoassays has evolved too. It resembles a detective assembling clues from a crime scene. The color change's intensity is gauged using a spectrophotometer, and the protein concentration is deduced by comparing it to a standard curve. Now, the advent of artificial intelligence has revolutionized this process, managing the colossal amount of data for more accurate and rapid analysis. It's like having a digital Sherlock Holmes, automating the process of mystery solving. So, ELISA, paralleled with its precision and sensitivity, is a keystone in immunology and disease diagnosis. New methods like multiplex immunoassays and the inclusion of AI offer even more profound insights into our body's intricate interactions. From detecting diseases, comprehending immune responses, to unveiling our biological system's secrets, these techniques play a crucial role in the grand narrative of life, helping us grasp the elaborate ballet of proteins within us. In this digital age where data is the new oil, bioinformatics software and protein databases have become indispensable tools in the study of proteins. These are like the vast libraries and the astute librarians of the biological world, helping us navigate the labyrinth of protein data. Let's start with protein databases. These are comprehensive repositories that store and share information about protein sequences, structures, and functions. Imagine them as colossal warehouses, each aisle filled with details about a different protein, ready to be explored. Databases like UniProt and PDB are filled with invaluable resources for researchers and scientists, serving as the encyclopedias of protein knowledge. Now, let's move on to bioinformatics software. These are powerful tools used to analyze protein structures, predict protein functions, and study protein-protein interactions. They're the analysts in our biological investigation, helping us make sense of the data. Software like BLAST, Clustal Omega, PML, and Cytoscape are the magnifying glasses and the deciphering codes in our pursuit of understanding life's blueprint. For instance, BLAST, or Basic Local Alignment Search Tool, is like a biological search engine. It allows scientists to compare protein sequences, helping them find similarities and differences. Clustal Omega, on the other hand, is a multiple sequence alignment program that aligns sequences together to find common patterns, like a puzzle solver aligning pieces to reveal the bigger picture. PML and Cytoscape visualize protein structures and interactions, acting as the illustrators of the protein world. With these databases and softwares, we can delve deeper into the world of proteins, uncovering their secrets and understanding their interactions. They are the librarians and the analysts in our quest to understand life's blueprint. Through them, we continue to unravel the mysteries of life, one protein at a time. We've talked about protein databases and bioinformatics software. Now, let's dive into the world of DNA sequencing, specifically Sanger sequencing. Developed by Frederick Sanger in 1977, this technique has become a cornerstone in the field of genomics. It played a crucial role in the Human Genome Project and continues to be an essential tool in various genomic applications. But what is Sanger sequencing? The Sanger sequencing method relies on the DNA chain termination principle. It involves enzymatic synthesis of complementary DNA strands in the presence of chain-terminating dideoxynucleotides or ddNTPs. Now, how does Sanger sequencing work? The process begins with DNA denaturation, where the DNA double helix is separated into single strands. A primer then anneals or binds to the DNA strand, and DNA synthesis begins. Here's where the ddNTPs come into play. When one of these ddNTPs is incorporated into the growing DNA strand, it causes the synthesis to terminate, producing fragments of different lengths. These fragments are then separated and visualized, allowing us to read the DNA sequence. Sanger sequencing has wide applications. It's used in genome sequencing, mutation detection, forensic analysis, phylogenetic studies, which is the study of evolutionary relationships, and microbial identification. The data analysis in Sanger sequencing involves base calling, where the DNA sequence is determined from the data, chromatogram analysis, sequence alignment, quality scores, and mutation identification. Despite the advent of next-generation sequencing technologies, Sanger sequencing remains a fundamental tool in molecular biology. Its reliability, accuracy, and cost-effectiveness make it suitable for various applications. Understanding the principles, working mechanism, and data analysis involved in Sanger sequencing is crucial for researchers and clinicians utilizing this technique in genomics and diagnostics. So whether we're decoding the human genome or identifying a new species, Sanger sequencing helps us uncover the secrets hidden within the DNA, one base at a time. Welcome to the next leap in our genetic adventure, the next-generation sequencing, often referred to as NGS. This groundbreaking technology has completely transformed the field of genomics, enabling us to perform highly efficient, massively parallel sequencing of DNA. Imagine being able to tune into 1 million TV channels all at once, with each channel broadcasting a unique genetic tale. But how did we arrive at this point? Let's take a retrospective look at how sequencing technologies have evolved. Traditional methods like Sanger sequencing had their limitations, such as high cost, low productivity, and strenuous procedures, which led to the invention of NGS technologies. NGS includes a variety of technologies that all massively parallel sequence, allowing us to examine millions of DNA fragments at the same time. Now we're going to explore 4 types of next-generation sequencing techniques. A. Illumina, Selector, sequencing. Based on the principle of reversible terminator chemistry and clonal amplification, this method involves ligating DNA fragments to adapters, forming clonal clusters on a flow cell, and carrying out sequencing by synthesis with reversible terminators and fluorescently tagged nucleotides. This technique is extensively used for whole genome sequencing, targeted sequencing, and transcriptome analysis. B. Ion torrent sequencing. This technique, which relies on semiconductor sequencing, involves amplifying DNA templates on ion-sensitive semiconductor chips. The release of hydrogen ions during nucleotide incorporation is detected. It is ideal for targeted sequencing, exome sequencing, and microbial genome analysis. C. PacBio single molecule real-time (SMRT) sequencing. This sequencing method is performed by observing the incorporation of labeled nucleotides during DNA synthesis. Single DNA molecules are immobilized in nanoscale wells, and polymerase kinetics are monitored as nucleotides are incorporated. This method is particularly valuable for de novo genome sequencing, long-read applications, and understanding structural variations. D. Nanopore sequencing. Sequencing is achieved by passing DNA through a nanopore, and the changes in current are used to identify the nucleotides. DNA strands are threaded through nanopores, and the anionic current are measured as nucleotides pass through. This technique is useful for portable sequencing, real-time monitoring of DNA, RNA, and detection of epigenetic modifications. NGS has unlocked new possibilities in genomics research, from identifying genetic variations linked to diseases to understanding intricate ecosystems. So as we venture deeper into the realm of NGS, remember, we are not just simply reading DNA, we're revealing the stories that define us as individuals. As we delve deeper into the world of next-generation sequencing, or NGS, let's explore the wide range of applications this revolutionary technology has to offer. A. Whole genome sequencing, WGS. In clinical diagnostics, WGS helps identify genetic mutations associated diseases, enabling early diagnosis and personalized treatment strategies. In population genomics, it aids in studying genetic variations across populations, providing evolutionary insights and tracing ancestral roots. B. Targeted sequencing. Exome sequencing focuses on the protein coding regions of the genome for a cost-effective analysis of genetic disorders. Custom panels target specific genomic regions for disease-specific research, speeding up the process of identifying disease-causing mutations. C. Transcriptome analysis. RNA-seq profiles the entire transcriptome, aiding in the understanding of gene expression patterns. Single-cell RNA-seq analyzes gene expression at the single-cell level, providing insights into cellular heterogeneity and disease progression. D. Epigenomics. DNA methylation analysis investigates epigenetic modifications to understand gene regulation and the role of environmental factors in disease. Chromatin immunoprecipitation sequencing, or ChIP-seq, identifies protein-DNA interactions, providing insights into chromatin structure and gene regulation. E. Metagenomics. In microbial genomics, NGS is used to analyze the composition and function of microbial communities in various environments, from the human gut to the deep sea. Virome and resistome analysis studies viral communities and antibiotic resistance genes, contributing to the fight against antibiotic resistance and emerging infectious diseases. F. Personalized medicine. In cancer genomics, NGS identifies genomic alterations in tumors, aiding in the development of targeted therapies. Pharmacogenomics uses NGS to understand genetic variations influencing drug responses, paving the way for personalized medicine. Now, let's move on to the essential part of next-generation sequencing: data analysis. A. Base calling. Quality scores are assigned to sequencing reads based on the confidence of base calls. B. Read mapping and alignment. In reference-based mapping, reads are aligned to a reference genome for variant calling. De novo assembly assembles reads without a reference genome for novel sequence discovery. C. Variant calling, SNP detection. Detection identifies single nucleotide polymorphisms. Indel calling detects insertions and deletions in the genome. D. Structural variant analysis. Structural variants are identified and their functional consequences are assessed. E. Functional annotation. Pathway analysis understands the biological pathways affected by genetic variations. Gene ontology analysis categorizes genes based on their biological functions. As we continue to harness the power of NGS, we're not just reading the code of life. We're using it to enhance our understanding of the world around us, to improve the quality of life, and to shape the future of medicine. As we continue to delve into the complexities of genomics, let's introduce a novel technique that is transforming our understanding of gene expression: spatial transcriptomics. This cutting-edge method combines spatial information with transcriptomic data, providing valuable insights into gene expression within complex biological samples. 1. The principle of spatial transcriptomics. The key principle involves capturing the spatial context of gene expression within tissues. This allows for the identification of gene expression patterns in specific regions, integrating traditional RNA sequencing with spatial localization information. 1. The procedure of spatial transcriptomics. The process begins with the preparation of tissue samples, which may involve sectioning tissues into thin slices or using intact tissues depending on the platform. A. Tissue capture platforms. There are two main approaches: in situ sequencing techniques, which sequence transcripts directly within the tissue sections, and spatially barcoded beads, which capture transcripts using spatially barcoded beads that are placed on tissue sections, allowing for subsequent sequencing. B. Library preparation. RNA is extracted from the tissue sections and reverse transcribed into cDNA. During library preparation, spatial barcodes are introduced to the cDNA to retain spatial information. C. Sequencing. High-throughput sequencing platforms are employed to sequence the cDNA libraries, generating data on gene expression and spatial distribution. D. Image registration. The spatial transcriptomics data is aligned with imaging data, allowing the correlation of gene expression with specific tissue structures or cell types. As we advance in our understanding of spatial transcriptomics, we're not only mapping the genetic landscape but also the physical geography of our genetic makeup. This integration of space and sequence is opening new avenues in our understanding of complex diseases, cell differentiation, and tissue development. The future of genomics is not just about reading the code of life in a linear fashion, but understanding it in its true 3D context. Journeying further into the world of genomics, let's pause to delve into the specifics of a revolutionary tool: cytoff, or cytometry by time of flight. This groundbreaking technology integrates the principles of flow cytometry with mass spectrometry, facilitating multi-parametric analysis at the single-cell level. Unlike the traditional flow cytometry that relies on fluorophore-labeled antibodies, CytoF utilizes antibodies labeled with unique metal isotopes, broadening the number of parameters that can be simultaneously analyzed, and thereby offering a more intricate view of our genetic makeup. Let's take a closer look at how CytoF can be applied in genetic studies. For instance, in single-cell genomics, it can be used to investigate cellular heterogeneity. Entails examining the differences among a cell population at the single-cell level, thereby providing insights into genetic variations among individual cells. Moreover, CytoF can identify and characterize genetic mutations within specific cell subsets, which aids in understanding clonal evolution and disease progression. In immune profiling, CytoF comes in handy in identifying cell subsets. It characterizes immune cell populations based on genetic markers, enabling a detailed immune profiling. It also assists in exploring the genetic determinants of immune cell functions and activation states, known as functional signatures. In the broader field of functional genomics, CytoF aids in gene expression analysis at the protein level, offering a complementary approach to traditional RNA-based methods. It can also assess the activation status of specific genetic pathways within individual cells, providing functional insights. When it comes to data analysis in CytoF studies, dimensionality reduction techniques like T-distributed stochastic neighbor embedding, TSNE, or uniform manifold approximation and projection, UMAP, are employed to simplify high-dimensional data for easier visualization. CytoF also helps in clustering and phenotyping, enabling the identification of cell clusters based on expression profiles and thus allowing for the characterization of distinct cell populations. Moreover, functional analysis is made possible by relating genetic markers to functional attributes through integrating genetic data with additional parameters like cytokine production or cell cycle status. On a broader note, CyTOF marks a significant stride in genetic studies. It equips researchers with the tools to delve deeper into the complex world of gene expression, cell differentiation, and disease mechanisms, thereby unlocking new possibilities for understanding and treating genetic disorders. As we venture further into the realm of genomics, the power of CyTOF will undoubtedly continue to shape our understanding of the genetic code. Embarking on our expedition into the realm of genomics, we delve deep into the workings of tandem mass spectrometry, a potent analytical method that is transforming the genetics field. This technique employs a two-step process of mass spectrometry to yield in-depth information regarding the structure, composition, and abundance of molecules. A sample preparation. We begin with extraction, where we pull out and purify the genetic material of interest from the biological sample. This could be proteins, DNA, RNA, or metabolites. If necessary, we may perform derivatization to modify the molecules to enhance their ionization efficiency or stability, particularly for metabolomic studies. B. Ionization. We move on to the next step, ionization. Here, an appropriate ionization method such as ESI or MOLDI is selected based on the nature of the molecules being analyzed. This stage, also known as the MS1 analysis, involves ionization of the molecules of interest to form charged species. The result? Large and complex molecules transform into ions, primed for further examination. C. Fragmentation. Post-ionization, we isolate the chosen ions from the MS1 analysis and subject them to fragmentation. This second stage, MS2, is orchestrated with the aid of fragmentation techniques like CID or ECD. This fragmentation process breaks down the selected ions into smaller pieces, offering a more intimate glimpse into the molecular structure of the original ions. The fragments born from the fragmentation step are then scrutinized in the second mass spectrometer. This mass analysis equips us with explicit information regarding the molecular structure of the original ions, shedding light on the intricacies of biomolecules such as peptides, proteins, nucleic acids, and metabolites. D. Data analysis. We proceed to the final step, data analysis. This involves searching the experimental MS spectra against databases to identify proteins, DNA sequences, or other genetic material. We then quantify the abundance of the molecules based on the intensity of the MS signals. As we forge ahead into the genetics landscape, tandem mass spectrometry emerges as a pivotal instrument. With its capability to furnish detailed molecular data, it is a building block in the field of genetics, assisting in the scrutiny of complex biomolecules. It is through methods like tandem mass spectrometry that we continue to progress our comprehension of life on a molecular plane. In this journey, we are going to delve deeper into the world of spatial transcriptomics, a groundbreaking field that is expanding our understanding of cellular complexity. This technique enables us to visualize and analyze the spatial distribution of gene expression within tissue samples, providing a new perspective on cellular function and organization. Our focus will be on the data analysis process in spatial transcriptomics, which encompasses several key steps. 1. Image alignment and registration. The process begins by aligning and registering the images from a tissue sample. This involves correlating the spatial coordinates of each gene expression event with the corresponding location in the tissue image. 2. Gene expression analysis. This stage involves analyzing the gene expression data from the tissue sample. Using advanced algorithms, researchers can identify patterns, compare gene expression levels, and gain insights into cellular function and complexity. 3. Spatial clustering. In this phase, clustering algorithms are used to group together genes that show similar spatial expression patterns, revealing the underlying structure of the tissue sample. 4. Pathway analysis. This involves studying the biological pathways that the identified genes are involved in. It gives insights into the functional roles of these genes and their potential implications in disease development. 5. Visualization, interpretation, and validation. The data is then visualized, interpreted, and validated using a variety of bioinformatics tools and databases. This step allows for the generation of comprehensive spatial maps of gene expression, opening new doors for biological research. In addition to the process, we'll also explore how spatial transcriptomics is being applied in various fields. It has been instrumental in studying tumor heterogeneity, enabling researchers to understand the genetic variability within tumors. In developmental biology, it provides a detailed map of gene expression during the formation of tissues and organs. In neuroscience, it's used to study the complex network of neurons and their intricate interactions. Finally, we'll also touch upon the challenges and future directions in spatial transcriptomics. Although the technique has proven to be revolutionary, it still faces hurdles in data analysis, interpretation, and applicability, which need to be addressed for further advancements. As we continue to refine these techniques and overcome challenges, spatial transcriptomics promises to bring a deeper understanding of the genetic orchestra that creates life. This scene will be enriched with video footage and illustrations to provide a comprehensive understanding. As we venture further into our exploration of genetics, we now introduce a new dimension of genomics: functional genomics. This multifaceted field focuses on understanding the roles, interactions, and functions of the various genes within a genome. The first part of our exploration will focus on gene function and regulation. We'll discuss functional annotation, a process that assigns biological roles to genes. This is done using a combination of databases, literature, and experimental evidence. We'll then delve into the study of regulatory elements, such as enhancers and promoters, which control gene expression. Next, we'll explore molecular networks and pathways. Here, we'll introduce the concept of interactomy analysis, which involves mapping the physical and functional interactions among proteins. This analysis provides valuable insights into the processes that occur within cells. We'll also discuss pathway analysis, which examines the involvement of genes in biological pathways, thus highlighting their collective impact. Finally, we'll discuss the functional consequences of genetic variations. We'll explore single nucleotide polymorphisms, SNPs. These small variations can have a significant impact on gene function and disease susceptibility. We'll also discuss structural variants, larger genomic alterations that can affect gene expression and function. This scene will be enhanced with animations and graphics to illustrate the complex processes involved in functional genomics. As we delve into this fascinating field, we'll unravel the dynamic aspects of gene expression, regulation, and the functional consequences of genetic variations, shedding light on the intricate dance of life at a molecular level. In this scene, we'll delve deeper into the tools and techniques that enable the study of functional genomics. We'll explore several key areas, each contributing unique insights into the complex world of genomics. A. Transcriptomics. We begin with microarrays, a hybridization-based detection technique used for profiling gene expression across thousands of genes simultaneously. Next, we journey into the world of RNA sequencing or RNA-seq, a high-throughput sequencing technique used to quantify and analyze transcriptomes, providing an unbiased and detailed assessment of gene expression, alternative splicing, and non-coding RNAs. B. Proteomics. We'll then pivot to proteomics, starting with mass spectrometry, a technique used for the identification and quantification of proteins based on mass-to-charge ratios. We'll also touch on two-dimensional gel electrophoresis, 2D page, a method used to separate proteins based on charge and size, enabling comparative protein expression analysis. See functional screens. Our journey continues with a look at RNA interference, RNAi, a method for silencing gene expression, and CRISPR-Cas9 gene editing, a revolutionary technique for precise genome editing. D. Epigenomics. We'll then delve into the realm of epigenomics, exploring DNA methylation analysis, a technique for assessing DNA methylation patterns to understand epigenetic regulation. We'll also cover chromatin immunoprecipitation, CHIP, a method for isolating DNA regions bound by specific proteins. E. Metabolomics. Finally, we'll touch on metabolomics, featuring mass spectrometry-based metabolite profiling, a technique for identifying and quantifying small molecules within a biological sample, and nuclear magnetic resonance, NMR, spectroscopy, a method for analyzing metabolite profiles. As we navigate through these techniques, we'll be using animations and graphics to illustrate complex concepts, making them easier to understand. This scene is a testament to the power and precision of scientific techniques, illuminating the intricate world of functional genomics. As we delve deeper into the world of genomics, let's explore how these scientific techniques are applied in studying plant diversity, creating climate-resilient crops, probiotic drinks, and GMOs for food safety and climate action, and revolutionizing the tracking and conservation of endangered species. First, we look at the role of genomics in plant diversity. Genomic techniques help in understanding the genetic diversity within and between plant species. This information is vital for biodiversity conservation, aiding in the protection and propagation of unique and endangered plant species. Next, we delve into how the study of genetics is transforming the tracking and conservation of endangered species. Using genetic techniques like DNA barcoding and genetic fingerprinting, we can now accurately identify and monitor endangered species, providing crucial assistance in their preservation. DNA barcoding, a method used to identify species using a short genetic sequence, aids in precisely cataloging biodiversity. On the other hand, genetic fingerprinting, a technique that involves analyzing the genetic makeup of an individual, helps in monitoring the genetic diversity of endangered species, thereby guiding effective conservation strategies. Further, we explore the development of climate-resilient crops. With the aid of functional genomics, scientists can identify genes responsible for climate resilience. This leads to the development of crops that can withstand harsh environmental conditions, ensuring food security in the face of climate change. We then move on to the creation of probiotic drinks. Genomic techniques are used to engineer probiotics with enhanced health benefits. By understanding the genomic makeup of these beneficial bacteria, scientists can optimize their health effects, leading to the creation of superior probiotic drinks. Finally, we discuss the role of genomics in the creation of GMOs for food safety. Genomic techniques can be used to create genetically modified organisms, GMOs, that are resistant to pests and diseases. This not only improves food safety, but also reduces the need for harmful pesticides. As we continue to harness the power of genomics, we're not just reading the code of life. We're using it to enhance our understanding of the world around us, to improve the quality of life, and to shape the future of agriculture, food safety, and species conservation. As we conclude this scene, let's reflect on the transformative power of genomics and its potential to revolutionize our world. Venturing forth in our exploration of genomics, we now delve into a captivating application: the resurrection of extinct species, or de-extinction. The idea, once purely the stuff of science fiction, is now within the realms of possibility thanks to advancements in genomics. Our journey begins with the role of next-generation sequencing in studying the genetics of extinct species such as the woolly mammoth and the Tasmanian tiger. These sequencing techniques allow scientists to decode the entire genetic makeup of an extinct species from preserved samples, providing a blueprint for their potential resurrection. From here, we spotlight the efforts made by various research institutes in resurrecting extinct species. This audacious task involves using gene editing techniques like CRISPR-Cas9 in combination with the knowledge of genetics. By splicing genes from extinct species into the DNA of closely related living species, scientists aim to recreate the traits that define these lost creatures. However, the pathway to de-extinction is not without its hurdles. We delve into the challenges and ethical considerations of such endeavors. These include the potential impacts on existing ecosystems, the welfare of the resurrected animals, and the question of whether our resources would be better spent on preventing current species from going extinct. As we wrap this scene, let's ponder on the transformative power of genomics. We're not just reading the code of life, we're using it to potentially bring back species that once roamed the Earth, opening up a whole new world of possibilities. The question we must ask ourselves is not just can we, but also should we? In this next scene, we'll be taking a deep dive into a topic that goes hand in hand with the power of genetic manipulation, the ethical dimensions of genetic engineering. As we navigate through the labyrinth of genetic possibilities, it's important to consider the moral implications of our actions. We'll start with the fundamental question: just because we can manipulate the genetic code, does it mean we should? This question takes us on a journey through the ethical minefields of genetic engineering, from the use of CRISPR-Cas9 to alter human embryos, potentially eradicating hereditary diseases, but also opening the door to the controversial practice of designer babies, to the resurrection of extinct species, which, as we've seen, could have far-reaching ecological implications. Next, we'll explore the concept of genetic privacy. With the rise of direct-to-consumer genetic testing kits, more and more people are sending off their DNA to be analyzed. But what happens to that data once it's been sent? Who has access to it? And how might it be used in the future? Then we'll delve into the potential for genetic discrimination. Could knowledge of a person's genetic predisposition to certain diseases be used against them by insurance companies or employers? Finally, we'll discuss the issue of genetically modified organisms or GMOs. While they hold the promise of increased crop yields and disease resistance, there's ongoing debate about their safety and impact on biodiversity. As we wrap up this scene, we'll reflect on the importance of these ethical considerations. While the power of genomics opens up a world of possibilities, it also presents us with profound questions about the kind of world we want to create. The genetic future is in our hands, but it's up to us to handle it responsibly. Welcome to Scene 26, where we will explore the burgeoning field of personalized medicine and the role genomics plays in it. Personalized medicine is a medical model that separates people into different groups, with medical decisions, practices, interventions, and/or products being tailored to the individual patient based on their predicted response or risk of disease. We start with a brief introduction to personalized medicine. It's an approach that integrates genetic understanding with clinical data to optimize and personalize patient care. It's a revolution in healthcare delivery, but it's only possible thanks to our advanced understanding of genomics. Next, we dive into pharmacogenomics, a branch of personalized medicine. Pharmacogenomics studies how genes affect a person's response to drugs. This field allows clinicians to design effective, personalized treatment strategies that minimize side effects and ensure maximum effectiveness. The scene then explores the role of genomics in disease prediction and prevention. With the help of genomic data, it's possible to determine an individual's susceptibility to certain diseases and conditions, allowing for preventive measures to be taken long before any symptoms appear. From disease prediction and prevention, we then move on to the role of RNA modification in disease. This is our fourth point. RNA modifications can influence gene expression and have been linked to various diseases. Understanding these modifications can lead to new therapeutic strategies. This will be followed by the fifth point, the role of small non-coding RNAs in disease. Small non-coding RNAs like siRNA, piRNA, and miRNA can regulate gene expression and have been implicated in various diseases. They hold potential as therapeutic targets. Then we'll delve into the potential for personalized medicine in cancer treatment. Cancer is fundamentally a genetic disease by mutations in the DNA within cells. Understanding a patient's unique genetic profile can help inform decisions about which treatments are most likely to be effective and which should be avoided. We'll also touch upon the challenges faced in this field. While the potential benefits are enormous, there are also significant hurdles to overcome. These include ethical issues, data privacy concerns, and the need for a robust regulatory framework. Finally, we'll conclude with a look at the future of personalized medicine. As our understanding of genomics continues to grow, so too does the potential for personalized medicine. It's an exciting that promises to revolutionize healthcare and lead to better patient outcomes. As we wrap up this scene, we'll reflect on the transformative potential of genomics in healthcare. The power of genomics extends far beyond the laboratory, impacting real people and their health every day. We're not just understanding life, but also improving it. Join us in the next scene as we delve further into the relationship between genetics and disease understanding. As we continue our journey into the world of genomics, we'll now explore how our understanding of genetics is revolutionizing the way we approach various diseases. First, we'll delve into the realm of hereditary diseases. These are conditions caused by genetic mutations passed from parents to offspring. By studying these mutations, scientists can predict the likelihood of these diseases manifesting, enabling early detection and treatment. From cystic fibrosis to Huntington's disease, understanding the genetic markers has been a game changer in their management. Next, we'll look at metabolic syndromes, a cluster of conditions that increase the risk of heart disease, stroke, and type 2 diabetes. Genetic factors can influence the development of these syndromes, and genomic research is helping to develop personalized treatment plans. By studying an individual's genetic makeup, doctors can tailor treatments to be more effective and have fewer side effects. Our exploration continues with viral diseases. The recent COVID-19 pandemic has highlighted the crucial role genomics plays in understanding the genetic makeup of viruses. Sequencing the genome of the virus has led to the development of effective vaccines in record time. Then we'll discuss functional characterization. By identifying unique molecular signatures in genetic diseases, doctors can improve prognosis and enhance the quality of life for patients. These signatures can also guide the development of new therapies targeting the underlying causes of the disease. As we wrap up this scene, let's reflect on the power of genomics. As we continue to unravel the mysteries of our genetic code, we're not just understanding life, but also improving it. The transformative potential of genomics extends far beyond the laboratory, impacting healthcare, biodiversity, and even our understanding of our own history and future. As we conclude our journey into the world of genomics, let's take a moment to reflect on the transformative power of genetic discoveries and the ethical considerations that come with it. Our exploration has taken us from the basics of genetics through advanced techniques in genomics to the pivotal role genetics play in understanding and managing diseases. We've seen how the power of genomics extends far beyond the laboratory, impacting healthcare, biodiversity, and even our understanding of our own history and future. But with great power comes great responsibility. As we venture further into the realm of genetic manipulation, we must grapple with the ethical implications. The potential to cure genetic diseases and enhance human capabilities is exciting, but it also raises questions about genetic privacy and the potential for genetic discrimination. Who should have access to our genetic data? How do we balance the benefits of genetic research with the need to protect individual privacy? And how do we ensure that genetic advancements benefit all of humanity, rather than widening the gap between the haves and the have-nots? Regulatory agencies worldwide play a crucial role in navigating these complex ethical waters. They set the rules for genetics research, ensure ethical practices, and protect genetic data. But these agencies can't do it alone. It's up to all of us, researchers, healthcare professionals, policymakers, and the public, to contribute to these discussions and decisions. As we step into the future of genomics, it's crucial to navigate this path with responsibility and respect for the power of genetic knowledge. The future is in our hands, and it's up to us to shape it responsibly. As we wrap up this comprehensive exploration, remember that our understanding of genetics is constantly evolving. The more we learn, the more we realize the vast potential of genomics. But let's proceed with curiosity, respect, and a deep sense of responsibility.